All right, well, welcome to our next module. This is basic genome-wide association studies, and this is one of the major workhorses of pretty much all genetic research these days. And we'd first like to thank our sponsors, the Computational Medicine at UCLA department, and they've provided money for the video cameraing and money for food as we film these modules. And so when we think about GWAS, we're trying to find a relationship between phenotypes and genetics. So when we think about a phenotype, there's a couple. Your phenotype is kind of your outward appearance of something. And it's a very loosely defined things because you can really have a lot of different things as phenotypes. There's kind of the very traditional ones of how tall is somebody, what's their BMI, what's their hair color, do they have diabetes, don't they have diabetes, do they have autism, not autism. However, you could also ask how much money do they make every year? This could also be a phenotype that you could measure. And you could ask, is there a relationship between genetics and how much money people make? Also, people's favorite color. You could consider that a phenotype. There's also these things, oftentimes endophenotypes, that are things like inside of us, such as blood sugar levels or perhaps a protein level. All of these can be phenotypes that we can then try to look for a link between what these values are and what people's genetics look like. So if we're measuring a phenotype, in this case sickle cell anemia, and we're also looking at somebody's genetics, say at RS334, which is a SNP. So one question we can ask is, does the genome impact the phenotype, or does the phenotype impact the genome? One of these two options is true, like 98% of the time, and then there's some edge cases we won't discuss. But in general, which one of these two possibilities do you think is the truth? Does the genetics impact the phenotypes, or does the phenotype impact the genetics?
Genetics.
All right, why? How do you get your genetics? From your parents, right? It's just like given to you, and then you've got it. And it doesn't change over time, right? Whereas, like, say, do you have diabetes or not? That is something that changes over time. So your genetics is fixed, and it's fixed basically, like, it's basically fixed for you your entire life. And so this principle that whatever happens in the genetics is not affected by phenotypes comes up to this idea called Mendelian randomization. And the idea is that you inherit your genetics from your parents randomly, not according to what phenotypes look like. And so we know that if we see something in genetics which is related to something in your phenotype, it must mean that something in the genetics is having an impact on the phenotype. And it cannot be that the phenotype is having an impact on the genetics. All right, so when we actually do this genome-wide association study, we're trying to figure out, okay, where in the genome is there an impact happening on the phenotype? So one way we can do this in the traditional way is called a case control study. And you got a bunch of people with a disease, say diabetes, and a bunch of people without diabetes, and you simply look at their genomes and you ask, where are they different? So in this case, they're looking at this variant right here, and there's this red allele and I guess this light blue allele that also exists there. And what you can see is that in cases, this red allele has this 58% allele frequency, whereas in controls it only has a 17% allele frequency. And so a researcher might conclude, well, cases have this a lot more often, and there's no reason they should have this a lot more often due to random chance. So it must be that whatever this allele is either itself or somehow tagging something else that does have an impact on disease risk. So you could be like, anyone who has this is just more likely to be a case than a control. And then a biologist can go in and instead of look at the entire genome to find out like what's giving rise to diabetes risk, they can just look at this little region of the genome and really like focus on that region because they know something there is giving these people a higher risk of having a disease. So this is a case control study. The other type of study you do with GWAS is called a continuous phenotype or a continuous trait. And height is the most common continuous trait, which is studied a lot. And that's basically because every single time we do any sort of study, one of the most important things people ask, not most important, but one of the most common things people ask is, how tall are you? And so basically every study that's ever been done, we have height data for that. So we're able to like use height all the time as a phenotype. And so when you do that on your y-axis, here we have a phenotypic score, but you could say how tall is a person. And then on the bottom, you have the SNP genotype. So this could be how many copies of your alternate allele do you have. So you could see if this were height, people who have no copies of the alternate allele are on average a little bit shorter than people who have 1 or 2 copies. And so as you carry more copies of the alternate allele, you become taller. And trying to understand this relationship between the genetics and the phenotype is what the GWAS is trying to do. So when we did the complex disease module, we kind of learned about this model of the genome. And when we do GWAS, we're going to basically assume a very similar model. So what we want to do is we want to estimate all of these betas. And in this, there's no hats on these betas. So this is the true betas and the true effects. And we really want to know all of them. The problem is we can't estimate what all of these are at the exact same time. And in statistics, we say we can't fit the model or we don't have power to fit the model. And the idea here is that there's just so many parameters. Remember, there can be up to 1 million SNPs, that if you try to regress height on 1 million different variables all at the same time, you're just not going to have power to have anything significant. So instead, what we do when we do GWAS is we fit what we call a marginal model. And so instead of regressing the phenotype on all the genetics at once, what we'll do is we'll regress the phenotype just on one variant position at a time. And then we'll estimate, so here we're regressing on the ith SNP, and so we're going to estimate what's the effect of the ith SNP on the phenotype.